Hello, my name is Nicholas Hanley, and today I'm going to discuss with you the molecular basis of muscle contraction. To start off, let's look at the objectives we're going to go over. First, I'd like to be able to describe the basic structure of actin and myosin molecules due to the fact that those are pretty essential to understanding how the whole muscle contraction process occurs. Secondly, we'd like to go over and be able to explain the sliding filament mechanism of muscle contraction, the big overall process of how the whole thing occurs. Next, we'd like to be able to describe the role of calcium ions and ATP molecules in muscle contraction. And finally, we'd like to be able to explain how rigor mortis and muscle relaxation occur. So let's start off by looking at actin and myosin molecules. Okay, the big thing here is thick versus thin filaments. The thick filaments are the myosin filaments here, as you can see in the center, and the thin filaments are actually our actin ones. Okay, so as we look at these, we're going to start by looking and talking more specifically about the actin molecules. So we're looking at these little helical shape where you have individual actin molecules that form a helical pattern as they attach to each other. You also have two other molecules involved with actin filaments. You have your troponin molecules, which are shown here in green, and the tropomyosin, which is kind of like a string connecting those green balls. So those three things together make up an actin filament. These are pretty important as we go through the size. If we look of the actin filament, the actin filaments are about 12 to 18. Or I'm sorry, the actin is 5 to 8 nanometers in diameter and about 1 micrometer in length. Also on this, troponin has what we call a calcium bonding site, which is going to come into play pretty importantly in a couple slides. With the myosin filament, myosin is the thick filament again here, and you see these little, we're going to call them the myosin heads coming off of them. Those are going to be important in the attaching to the actin molecule to actually propel the muscle fibers inward to contract and release to relax. The myosin filaments are 12 to 18 nanometers in diameter and about 1.6 micrometers in length. So myosin, these, like I said, the heads here kind of looks like a golf club or intertwined golf clubs. There's two heads that stick up in each place, and those are going to play a big important role in cross bridge formation. So they're going to connect to our actin molecules, as you can see in a couple places here. It's doing that. What's unique about the myosin molecules is they have a couple hinge joints. They have one on the tail that will bend slightly to allow the molecule to attach to the actin site, and then the heads can actually shift on their hinge point, and that will actually push the actin filaments down towards the center of the muscle fiber. Okay, so the sliding filament mechanism or sliding filament theory is the main explanation for how this process works. So if we look, the first thing I want to point out is that our myosin molecule, our thick fiber, is actually going to be stationary. This is where the cross bridges bind the actin, and they're going to pull it inward. So the H zone here is where we have the myosin band. Actin actually overlaps the myosin fiber on two sides here. So this is an actin molecule, and it's overlapping here, and this one's overlapping on this one. So it does overlap the myosin, and the length between two two myosin molecules right here, where you see it's just actin filaments, there's no myosin involved, that is our I band. The I band is actually going to shrink or decrease in size as the muscle contracts, because these actin filaments here are going to squeeze towards the center of the H zone, and that actually limits or reduces the space in this I band. The A band is the myosin band, or the length of the myosin molecule, and again, those do not contract or move, they're stationary. The thin filaments, our actin filaments, are what's going to actually move. But regardless of what we're looking at, in both of these cases, neither fiber actually decreases in size. They just overlap more or less, and that's what reduces or contracts the muscle fiber. Okay. If you look at how a muscle actually contracts, how this actin and myosin actually interact with each other, we're going to come to a thing we call the power stroke. That's what actually moves the actin along the myosin fiber and shrinks or decreases the length of the sarcomere or muscle cell in general. Okay, so the big thing we have to know here is we get that on the actin molecule you have the troponin molecule that has a calcium binding site. Okay, so here calcium binds to that troponin and it causes the troponin molecule to change its conformation. So when it does this, it kind of pulls the tropomyosin out of the way as well. And by moving both of these pieces, it reveals the myosin acting or active site there to allow the bonding to occur. Okay, so we see here cross bridge is attached to the actin. We see that when the binding site's open. Once myosin binds, there's these hinge joints that are going to come into play. So if we look, the myosin is actually going to, the head of the myosin is going to hinge at about 45 degree inward, and that stroking motion is actually what's going to pull the actin filament closer towards the center of the sarcomere. So each time we do this, and it is a cycle because once we're done, we can reset and we can do the process over again. But each cycle, each movement here only moves the actin filament a slight bit. So to get full contraction, we have to do the move through the cycle several times. So it's kind of like when you're pulling a rope hand over hand, you're grabbing and pulling, and then you release it and grab another place and release and pull. You keep doing that. The other thing that I think is unique about this is your myosin filament, your thick filament, is surrounded by 6 different thin or actin filaments, and all 6 of these actin filaments are moving up and down the myosin filament as we're doing this process. They may not be they do it simultaneously, but they may not be moving exactly in the same sync. So 3 of the fibers might be actively moving while the one is kind of paused, waiting to reload to go through the cycle again. So there's a little bit of flexibility there. Okay, next we're going to look at the parts of the muscle fiber that actually make this happen. So calcium is important. We're going to see how it gets to the cells to make this work. Overall, skeletal muscles are stimulated to contract when a motor neuron releases acetylcholine into the neuromuscular junction, and that's going to cause an action potential to occur in the muscle cell or in the sarcolemma. So what we need to do is be able to conduct that action potential to the muscle cells so they can do their job. So one of the things that's going to do this are these transverse tubules, and you can see them here moving down here. So they're perpendicular to the surface, and they're perpendicular here, and they extend down into the surface to the interior. And their job is to continue. They are continuous with the sarcolemma, and they will pass that action potential from the surface down into the T-tubules, and that rapidly transmits the surface electrical signal into the interior of the fiber. So there's these other muscle fibers in here that need to get that attention as well. So what this does, an action potential that makes it into the T-tubule actually causes a permeability change in the sarcoplasmic reticulum, the SR, and that's this mesh-like or net-like substance here between them. So T-tubules, sarcoplasmic reticulum. We change the permeability in here, and it makes things go a little faster. It makes the nerve impulse go so we can cause this contraction to occur. And again, we're back to calcium. Okay, so the sarcoplasmic reticulum surrounds the myofiber, like we said, like a netting or a netting. And there are these lateral sacs that are at the ends of them that store calcium ions. So as the action potential travels through that, when it reaches the sarcoplasmic reticulum, that triggers these lateral sacs to release the calcium ions. And when these calcium ions release, then that triggers the muscle response, the contraction. Um, as we look at these, um, there's a dihydropyridine receptors found in the T-tubule membranes, and they're membrane proteins, and they serve as voltage sensors. And basically, if depolarization is going to activate these sensors, these receptors, and that's going to trigger foot proteins, calcium receptors or ryanodine receptors in the lateral sacs. It's going to trigger those to function. Now, there is a small gap between the T-tubule membrane and the lateral sacs, and that trigger is what happens here. This foot proteins, the dihydropyridine receptor bridges that gap to allow that signal, that information to pass between the two. And it also does serve as a calcium release channel. So as we release calcium ions, it gets exposed to the binding sites on actin, those troponin molecules, and that causes that conformation change to open up or expose those actin binding sites so that we can connect the myosin and the actin together. Okay, now ATP is a pretty important energy carrying molecule in cells. If we look at a myosin head here, there's actually two sites. There's an actin binding site, and that's what's going to connect to the actin or the myosin binding site on the actin molecule. So those two sites connect to each other. But it also has an ATP binding site on the head, and that's going to be important. Okay, we need to have that. So what it does, it connects to an ATP molecule, and when the ATP connects, it actually decomposes or breaks it down into ADP and inorganic phosphate. Okay, so when we do that, that stores some energy in the bond of myosin. It kind of the analogy they give in the book, you're basically cocking the gun, getting it ready to go. So we want this myosin head to be able to hinge and propel or do the power stroke to move the actin filament. ATP provides the energy to make that work. So when the muscle gets excited, calcium pulls the troponin complex away. Binding site allows the energized myosin cross bridge to form and attach to the actin molecule. That the actual physical act of the myosin and actin bonding together causes us to pull the trigger of this loaded gun, and that's what allows the myosin head to bend and produces the power stroke, which moves the actin filament along the myosin filament towards the center. When we do this during the power stroke, the inorganic phosphate gets released, and then after the completion of the power stroke, the ADP molecule gets released. Once those two things are released, the ATP binding site here is now open again. So it could accept another ATP molecule. This is important because until another ATP binds to it, the actin and myosin are going to stay bound together. So we need ATP in order to let go of this bond. Once ATP binds, again, it returns myosin to its unbent form, so it dehinges, and then a new ATP can be split again, reactivating the myosin or recocking the gun and getting everything ready so that we can go through another power stroke. So that's cyclical manner of this. Rigor mortis is something that happens. It's the stiffness of muscles that occurs after death. It's related to this calcium and ATP stuff, so we're going to discuss here. It typically starts about 3 to 4 hours after death and lasts up to about 12 hours. That is slightly dependent upon environmental conditions. Different temperatures and things can factor into that. But again, from the last slide, remember, in order for myosin and actin to release from each other, we need fresh ATP. However, when an individual dies, an organism dies, we tend to see the calcium levels, the ion levels, are going to increase. And that can be because there's leakage of the calcium ions from the lateral sacs. And our extracellular calcium ions could also leak in because obviously the cell membrane is no longer functioning like it should. It's not being actively monitored there. So this increase in calcium ion levels um, does what it's supposed to. It removes the troponin and tropomyosin complex from the binding sites and allows actin and myosin to stick together or to bind. Now, what's unique here is once that bond forms, we cannot remove it or break that bond unless we have ATP available. And dead cells do not produce any more ATP. So once we bind together, we are locked in that rigid position. Um, so the thick and thin filaments stay linked together, and that's why the muscles have that stiffness to them. It will eventually subside as the proteins that run the process that keep the muscle contraction working are eventually going to decay, and that's why the body eventually does relax again. Okay, muscle relaxation. Okay, we don't want our muscles to always be tensed, and if calcium kind of initiates this stuff happen, the removal of those calcium ions is going to allow the muscle to relax. So when we return that calcium to the lateral sacs and no more action potentials are being sent telling us to contract, then we'll go back. There's a special pump. It's called the sarcoplasmic endoplasmic reticulum calcium ATPase or SERCA pump that's going to actively pump the calcium back into the lateral sacs for us. So pretty important role there. Again, as long as the end plate or the action potential is being produced, acetylcholine and all that stuff is still working, and the muscles getting that signal to contract. Once that action potential stops, then we can start the process of removing these calcium ions through this SERCA pump. And we remove calcium, that causes the troponin molecule to change its shape back to the original one, and the tropomyosin and troponin then move back over the active binding sites on the actin filament and prevents the myosin from being able to bind. So this is how we get those thin filaments back to the resting position. Duration of muscle contractions is if we compare muscle contractions to nerve impulses or action potentials, the duration of muscle contractions is much longer. Okay, and action potential lasts normally 1 to 2 milliseconds, as you can see in the bottom graph here. But a muscle contraction can be anywhere from 30 to 100 milliseconds, depending on whether it's a short twitch or or fast twitch or fast acting or a longer acting muscle type. So we can see a little bit of variance there, but regardless, 30 milliseconds is still a lot longer than 1 to 2 milliseconds. So if we look at this contraction is going to lag behind the impulse, the action potential, because we got to get everything set up and make sure we're getting all the excitation, contraction, coupling reactions ready to go before we can actually go and contract. So this little lag time we have is called the latent period. Okay, so it's the time delay between stimulation and the onset of the contraction. So you can see that in this first part. Next, we also need time to generate tension within the muscle fiber. So as we slide the thick and thin filaments through cross-bridge activity, we're building tension. And the maximum tension occurs at the peak here. So that's this top, or maximum contractile response. So this contractile time is the time it takes to start contracting to the tip or the most aggressive that it's going to be. So until peak tension occurs or develops. So this takes, on average, about 15 to 50 milliseconds, depending on the muscle fiber we're working with here. And then our contractile response finalizes when the lateral sacs have taken up all the calcium ions again. So reuptake of these calcium ions takes a little bit of time. So relaxation time is the time from the peak tension until we've accomplished the fact of bringing all those calcium ions in, and we're complete, we're done, and the muscle is back to its resting phase. And again, this can take between 15 and 50 milliseconds depending on the muscle cell type again. So overall, those times, again, 1 to 2 milliseconds for an action potential and 30 to 100 plus milliseconds for a muscle response are pretty important to provide muscle contractions of varying strengths. So being able to have that gap in time allows us to analyze that and work with those things. Um, Finally, we're going to talk a little bit about muscular dystrophy here. Muscular dystrophy is a genetic condition in which obviously there's muscle weakness and various things that come into play with this, but the muscle in the body actually gets or degenerates, and it's not regenerated. It forms things like scar tissue and fibrous tissue instead of muscle tissue. So you can see it starts this way and it progresses to where degenerates the muscle tissue. Obviously results in muscle weakness in the patients, and the severity of muscular dystrophy actually varies from patient to patient, even with the same genetic condition. So because we see variety within the same gene mutation, the severity is kind of controlled by maybe other genetic factors that factor in there, as well as environmental things. So that's why we see various forms there. One of the potential treatments that we're looking at now is with muscular dystrophy, the transforming growth factor beta, or TGF beta, has been shown to be upregulated in in muscle cells going through dystrophy. So what we're trying to do there is, if we can figure out a way to alter the TGF beta pathway or even modulate the activity, we may be able to alleviate some of the symptoms of the condition of the dystrophy and try to bring back some better results. So just to review, we wanted to describe the basic structure of actin and myosin molecules, and I think we were pretty successful in that, to understand what goes on when muscles contract. We talked about the sliding filament mechanism, how the actin molecules walk up and down the myosin filaments as the power strokes occur. We described the role of calcium and ATP molecules in contraction. The calcium is what opens up or exposes the active sites on the actin molecules, so myosin can grab it and move it. And ATP, without ATP, we would never be able to release the myosin actin bond, so we would be stuck in that position, and that would not be a good thing. That's what leads to rigor mortis. And then muscle relaxation, the big thing there is if calcium kind of provoked the muscle contraction, removing the calcium from the environment will allow the muscle to go back to its relaxing or resting phase. And here are my references, and thank you for your time.